Hi there, everybody. This is Cellular Respiration video lecture video number 3 right here. Now, this video today, this video is going to focus on specifically the path that leads towards anaerobic respiration. So we're going to first go down the anaerobic pathway to explain to you what's happening in the anaerobic pathway. The lecture is going to focus on pyruvic acid entering this pathway and what happens. And we're going to talk about two types of different fermentation. You may have heard of this term fermentation before. It's generally linked to the process of making alcohol, alcoholic beverages and things of that nature, wine or beer or the different types of liquors that they are produced by using bacteria and yeast and having them go through fermentation, alcohol fermentation. And they produce alcohol that way. That's the way it's done. In order to do that, they have to remove the oxygen. It's an anaerobic pathway. And anaerobic means no oxygen. Okay. So our last lecture focused on glycolysis, which happens first. It's the first part of cellular respiration. So we'll go over that real quick. Glucose gets broken down or oxidized and it gets turned into pyruvic acid. During this process, an electron carrier picks up electrons and NADH is formed. This is an important electron carrier. We'll talk about its function in a later lecture. And during glycolysis, 2 overall ATP are produced. Excuse me. All right, so we're going to talk about pyruvic acid entering 2 possible types of fermentation pathways. And if you notice it, 2 ATP are made in glycolysis, but 0 ATP are made in fermentation. But fermentation does have a job. Its goal is not to produce ATP because it's not producing much ATP. The goal of fermentation is to produce NAD pluses, electron carriers. Why? You got to think of this as a cycle that it doesn't want to stop. By producing NAD pluses, the cell can now go back and continue doing glycolysis. So the whole purpose of fermentation is to make NAD pluses and keep glycolysis going. Organisms that can live doing just anaerobic fermentation, well, they're not living, they're not moving, they're not doing a whole lot of stuff. Their overall ATP production is only 2 ATP. If you compare it to the aerobic pathway of 38 ATP, it's not a very efficient path. No, what happens is pyruvic acid gets broken down these fermentation pathways, and it doesn't yield any more ATP than the original 2 that were made in glycolysis. So this total 2 came from glycolysis. Okay, so now I'm going to go in detail on the different types of fermentation. And once again, the whole goal of fermentation is to keep making NAD pluses so that you have electron carriers to aid and facilitate glycolysis. So fermentation, all right? So now we're going to talk about the reduction, all right, of pyruvic acid. All right, so now pyruvic acid, it's going to be broken down in a different type of set of reactions. It's not going to be oxidized. It's going to be reduced, actually. And it's going to be broken down in the absence of oxygen. Fermentation means oxygen is not present. So this is the anaerobic pathway. All right. What does pyruvic acid get turned into? I'll explain that all using pictures in a second. These are the products that you end up that are being produced. And I already mentioned to you the whole purpose of fermentation or anaerobic respiration is to produce NAD pluses from those NADHs that were made earlier in glycolysis. So the cycle needs to keep going. Fermentation also takes place in the same location as glycolysis. It happens in the cytosol, all right, and 0 ATP are made, as I explained to you already. All right, so here's the first one. This is lactic acid fermentation. So glycolysis takes place. Pyruvic acid is made. During that step, NAD pluses are going in and NADHs are coming out. Electron carriers are there. They're produced. 2 ATP are made during glycolysis. Then pyruvic acid goes through another set of reactions, okay? It gets reduced. How do we know it gets reduced? Well, look, NADH goes in and NAD plus comes out. So NADH gets oxidized. Therefore, pyruvic acid is now going to be reduced. All right, so it's being changed and tweaked. And during this reaction, this reduction, pyruvic acid is becoming lactic acid. All right, lactic acid. This basically is what we would end up having in our muscle cells if we were exercising and we got really tired and fatigued and ran out of oxygen. Our body would shift in temporary moments into lactic acid fermentation. And this is the chemical in our muscles that would make them feel sore and achy and even burning at times. That's the lactic acid. That's our muscles shifting to anaerobic respiration because they ran out of oxygen temporarily or for moments. Now, this type of fermentation is also used to make cheese. Special bacteria are involved in that. So yeah, when you eat cheese or any dairy product, you're eating lactobacillus bacteria. So they say the bacteria you find in yogurt are good bacteria. They help your gut do well. Okay, so in this case, look, back again, let's look at this. What is the whole purpose of this whole setup? Well, pyruvic acid is broken down in a different set of reactions. And the whole purpose is to get these NAD pluses back to glycolysis to keep this process going. So it's basically a cycle that the cell does not want to stop. And in order to make sure it doesn't stop, it needs to recycle back these NAD pluses back to glycolysis. Glycolysis turns them into NADHs, and the NADHs help facilitate this part, this lactic acid fermentation. Now, it occurs in our muscle cells. It occurs in some bacteria, some yeast. Those, like I said, they're used to make these different types of dairy products. Okay, all right. And I already said to you, this is what makes your muscles hurt. This is what makes them sore. The next kind of fermentation is called alcohol fermentation. Now, we do not do this, right? It doesn't happen in animals. It's happening in bacteria, in yeast, and those types of organisms, and some plants. So once again, glycolysis happens first. 2 ATP are made, NADHs are made, pyruvic acid is made. After that, this is done by different organisms, not the other previous organisms. So it's a different set of enzymes are responsible for breaking down this pyruvic acid. So now it's reduced and it's turned into ethyl alcohol, right? During that process, so if you notice 3 carbons, now we have 2 carbons. Well, one of those carbons had to go somewhere and it's released right there. There's that carbon right there being released. Okay, now, once again, just like the previous process, NADH goes in, NAD plus is recycled back to keep glycolysis going and keep this process happening over and over again. The main purpose is to make NAD plus. So ethyl alcohol would be a waste product of this particular process. All right, so quick recap, glycolysis happens first, NADHs are made, then NADHs are used. They go in, NAD plus comes out, they're oxidized in this process. Okay. Now, when is this used? Well, people use this process when they're baking. When people are baking, they want carbon dioxide gas bubbles to be put in things like bread, so you can make bread or cake rise. So when they're using yeast, the yeast are buried inside of the dough. The oxygen runs out pretty quickly, so they're now anaerobic. And you put them in the oven and you cook them for a while, and they do anaerobic respiration at certain temperatures where they're functioning, and they are producing CO2 gas. It makes your bread rise. It makes your cake fluffy and your dough not so tough and dense. So they're used, specifically yeast are used in the production. But by the way, everyone said, well, that means they produce alcohol. Well, the alcohol evaporates. It's gone. I'm just showing you the process. This is air bubbles. Those are CO2 bubbles in the bread here. And these are showing you different types of fermentation, what can be produced in all the different kinds of fermentation. Okay, so this is ethyl alcohol, this is lactic acid, different kinds of cheeses or different kinds of bacteria are doing that. Acetone, isopropanol, different type of bacteria are involved or different organisms, different species. Vinegar is done this way also. And how efficient is anaerobic respiration? Well, we just said you end up with 2 ATP, comparing it to 38 ATP on the other side. So 2 ATP is not very efficient. It's considered 2% efficient at extracting energy. So 98% energy lost as a result of just anaerobic respiration. So you're not getting much energy out of the sugar that is going into it. Which organisms can survive doing this? Unicellular ones, small one-celled organisms that are probably not moving a whole lot. And if they're multicellular, they're definitely going to be not moving a lot. Okay. And organisms that do it, for example, we can do it, but we would do it in shorter windows of time. If we stayed anaerobic, we would die. And that will mark the end of this lecture.